Started. So we're going to spend 2 lectures on ligand-target interactions. And today it's going to basically be the basics of ligand-target interactions so you can become familiar with it. And then on Friday, we're going to go through a very important concept in pharmacology, which is selectivity. So as you kind of surmised from my first lecture on Monday, drugs are essentially ligands, and I want to stress that ligands can interact with any one of a number of targets. We talked about various types of receptors and even enzymes, other things like that. But remember that the ligand is what binds to the drug target, but bear in mind that there's probably also an endogenous ligand. So for instance, the estrogen receptor. Ordinarily, what binds the estrogen receptor is estrogen. But drugs have been developed that bind that same receptor. And so most often in pharmacology, you're going to have some kind of a competition with an endogenous ligand, or you might even prevent the binding of an endogenous ligand if you block a receptor. So bear that in mind, that what we say today and Friday, when we talk about ligands, the principles apply to both exogenous ligands, which are drugs, or the endogenous ligand. And so all this stuff applies to both cases. Okay, so let's talk a little bit about ligands. So let's see here. A ligand is defined simply as a substance that forms a complex with a biomolecule that's in your body. Usually it's a receptor or an enzyme, for instance. And what happens is the following. We're going to start to talk about this a little bit in terms of some kind of light chemistry because we want to be able to describe this numerically. But basically the essence of what happens is here's a receptor in your body. Let's say that's the estrogen receptor. Here's a drug that binds it. So you have a free ligand and a free receptor, and they can get together and form a ligand-receptor complex. And there is a rate constant that can describe that association, K1 in this case. Now remember, those ligand-target interactions are usually non-covalent in pharmacology. So just as this can associate, the ligand and the receptor can get together and form a ligand-receptor complex, this can also dissociate, okay? And that's this other constant K2 here. So remember, this is kind of a bimolecular interaction, and there's always association going on, and there's always dissociation going on. But what's going to determine how much of this receptor is occupied by the ligand is going to be basically the affinity of that receptor for a particular ligand. And that can be very strong or it can be very weak. It can vary over several orders of magnitude depending on what drug you have. So in terms of pharmacology, we can think in general of different types of action of the drug, the ligand. There are agonists that when they bind the receptor, they activate that receptor, and then whatever is downstream from that receptor is going to happen. We also have antagonists that can, when they bind the receptor, they prevent that receptor from functioning. And something rather similar, inhibitors, which might actually sometimes even irreversibly inhibit the receptor because they bind so tightly. Or they maybe change the conformation of that receptor such that it's impossible for it to bind its endogenous ligand. And in the case of enzymes, the ligand is actually a substrate. So if you recall from how enzymes work, a substrate gets together with an enzyme, and the substrate is the chemical entity that the enzyme acts on, that it catalyzes, and it will turn the substrate into products. And so you can actually inhibit enzymes by coming up with drugs that look like the normal substrate of the enzyme. Okay, so little bit more about ligands. For receptors, ligands, the drugs are typically agonists or antagonists. They activate or they inhibit. For enzymes, we have substrates. That's the endogenous entity that that enzyme acts on. And then we have inhibitors, so we can inhibit enzymes. But basically, anything that we can measure an affinity for, we can use ligand target science to figure it out. Okay, so let's talk a little bit about agonists and antagonists. So here's a cell, here is a receptor, and when the ligand binds the receptor, it can fully activate whatever function is downstream from that receptor. Maybe it causes the cell to secrete some substance, for instance. So an agonist typically will, that's the definition of agonist, is that it will act, is something that will activate a receptor, okay? If you have an agonist alone, you're going to get full activation. An antagonist is something that inhibits the receptor. So it can occupy the receptor, but it doesn't activate that receptor. So it kind of blocks it, right? So when agonist alone is present, that receptor is disabled and you don't get any corresponding function out of that cell. So an interesting thing happens when you add an agonist and an antagonist together at the same time. Both of them have an affinity for the receptor, right? If the agonist binds, it activates. If the antagonist binds, it blocks. But if you have both together, that sets up a competition. And so you'll get partial activation under those considerations. Now, this is oftentimes the real story of what's happening in pharmacology, because you have a drug that's been introduced into the body that's competing for a receptor along with whatever the endogenous ligand is. And you can think that if there's more drug, probably you'll get more inhibition. If there's less drug, you'll get less inhibition, because the competition becomes unfair, right, when there's a lot more drug present than the endogenous ligand. But each of these, the agonist and the antagonist, might have different affinities based on the chemistry as well. And that's also going to affect the degree of inhibition here, okay? So we need to determine, obviously, some kind of a quantitative way to look at how much the affinity, how big the affinity is, or how small. And we'll get into that today. Now, for enzymes, things are a little bit different. So here's just a normal enzyme working on its normal substrate. Remember, the substrate is the thing that the enzyme acts upon. So the enzyme binds the substrate, and then an enzyme is a catalyst, so it catalyzes some chemical reaction that occurs within the substrate, in this case a cleavage reaction, and the substrate is cleaved into two different molecules, okay? So the place on the enzyme where that occurs is called the active site. And so here comes the substrate, binds to the active site. You have an enzyme substrate complex, then catalysis occurs. In this case, you get a cleavage event. Now, you can actually inhibit enzymes by using an inhibitor. So an inhibitor has the property that it can occupy the active site, but it cannot be acted upon by the enzyme. In other words, it can't be metabolized. And so this enzyme is now taken out of action. If a substrate comes along, it's blocked. It can't get to the active site because it's already occupied with an inhibitor. So kind of similar to what goes on with receptors, but kind of different. So ligands can be all types of things. They can be small organic molecules. And there are many endogenous ligands that are small organic molecules. But they can also be drugs. In this case, here is nicotine binding to a receptor. And you can see in a space-filling model what that interaction looks like. But ligands can also be proteins or peptides. So for instance, in immunology, T cells can become activated when they interact with B cells. But what happens here is that on the T cell, there's a T cell receptor, and it can interact with an antigen-presenting cell right here. And this is basically a protein-protein interaction. And on the antigen-presenting cell, there's a protein called an MHC protein, major histocompatibility complex, and it presents peptides that have been made in the antigen-presenting cell. So for instance, it might be presenting an antigen, a piece of a protein from an infectious agent, and then once that's displayed on the surface of this cell, it can interact with the T cell receptor and activate this T cell. So this is a case where the ligand is a combination between an infectious disease-derived peptide and an MHC molecule. Ligands can even be gases. So for instance, this is something like hemoglobin here. And there's an iron atom that can undergo redox reactions here, and it can bind oxygen under certain conditions. So that's an example of a ligand being a gas. So all sorts of things can be ligands. So now let's get into a little bit more pharmacology here. So this is the structure of estrogen, which is also in some parts of the world called estradiol. Sometimes it's spelled O-E-S-T-R-O-G-E-N, if you're in England, for instance. But it's a sterol structure, and that's estrogen. Well, here is an antagonist drug for the estrogen receptor, raloxifene. And you can see that it doesn't look exactly like estrogen, right? But it has some features in common with estrogen. For instance, it has this kind of purine-like group here, but it doesn't have, 4 interconnected rings, but it has a lot of aromatic ring structure to it. So it's similar, but a little bit different. And both of these entities, estrogen and the endogenous ligand, and an antagonist drug, raloxifene, they can both bind an estrogen receptor, but they do so differently. So let's get into what the differences are. So this is a crystal structure of a portion of the estrogen receptor, shown in red and blue. And here is estrogen here. And what you can see is that there's a certain conformation of the receptor when estrogen is bound. But up here, you can see that, and actually, this is actually the estrogen up here, but there's a critical helix called helix 12. That's actually part of the estrogen receptor. Actually, I think this is estrogen down here. But what you can see is that the take-home message is this overall estrogen receptor has a certain conformation. This is being activated because it binds estrogen here. But when you add the drug raloxifene, you can see that the position of some of these helices within the receptor change, and the overall structure of the estrogen receptor is a little bit different when it binds raloxifene as opposed to when it binds estrogen. And in this case, it's a configuration that prevents that receptor from functioning. So raloxifene acts as an antagonist. Here it is shown in a slightly different way. Here is the estrogen receptor, and we're just kind of highlighting specific amino acids within the estrogen receptor. And here is estrogen itself, and you can see that it interacts with certain amino acids in the estrogen receptor. Histamine 524, glutamate, arginine. You can also see that even a water molecule is involved in this interaction, interestingly enough. But here is the same receptor, but now the binding site, the receptor ligand binding site, is occupied by raloxifene instead. And you can see that the position of a lot of amino acids are quite different. This funny little tail that hangs off the end of raloxifene is altering the structure of the receptor. So they both estrogen and raloxifene have an affinity for the receptor, but the structure of the receptor is different when the drug is bound to it, as opposed to the endogenous ligand. Questions on that general concept? So the receptor itself can change structure to kind of an abnormal sort of situation when certain drugs bind. So more about ligand-target interactions. Once again, targets can be proteins. There are even targets that can be nucleic acids. So for instance, there are drugs that bind nucleic acids. We're going to talk about some of them, like DNA and RNA. But the interaction between the ligand and the target can be very long-lived or very transient, okay? Because remember, this is a chemistry thing. There's association and dissociation going on. So what do you think? If the affinity of the ligand and the target interaction is very high, what do you think the half-life of that interaction is going to be? Take a guess. Say the affinity is very high in one case, but it's very low in another. If the affinity is very strong, that's going to tend to stick together, right? So you're going to have a really long half-life of the ligand-target interaction. If the affinity is very weak, then dissociation can readily happen, and the occupancy of the receptor for the ligand will go down. So what controls the strength of the interaction? The affinity is basically the strength of the interaction. Well, it's basically controlled by intermolecular forces. Remember, these are not covalent interactions. It's not covalent binding, it's non-covalent binding. So we have things like the charge difference between the ligand and the target. Other types of interactions, hydrogen bonding, that type of thing. Also the size, the general shape of the ligand binding pocket on the target is very important. So that's sterics, size and shape. Okay, so you see this little end of this molecule kind of fits in with this end of this molecule, kind of like a lock and a key. And that steric configuration is very important for the interaction. So ligand-target interactions are indeed a kind of bonding, but it doesn't involve the formation of covalent bonds. Rather, it's non-covalent. And so we're talking about weaker molecular interactions. So here are some of these non-covalent interactions we're talking about. Ion-dipole interactions, hydrogen bonding, dipole-dipole, ion-induced dipole, dipole-induced dipole, and also even van der Waals forces, which are, but this is in the order of the strongest to the weakest. And so you can see, for instance, here are ion dipole, here's hydrogen bonding, dipole. You can see what they all look like. But these tend to be very strong and these are weaker. Okay. So let's now get into a little bit of the biochemistry of ligand-target interactions. So I've been talking about, affinities, how tightly a ligand binds to a target. We can think of this, there's actually some crossover between just classic chemistry. So this is the Gibbs free energy equation here. And here are, we'll just go through this quickly. It's not too tough. So delta G0 is the change in free energy. Delta H is the change in enthalpy. And T is temperature. And S is entropy. So there's a term in this that is temperature dependent, and it has to do with the entropy as well. So what are these things? Well, a lot of people let's start with entropy. A lot of people understand entropy. That has to do with the randomness of the system. the disorder of a system. So a neatly stacked pile of bricks has more entropy than a random pile of bricks. Enthalpy, though, is a kind of a chemistry term. And I think the easiest way to understand that is it's the total bond energy in a particular system. And I'll show you an example in a moment. So anyway, you have this equation where this is going to, the delta G0 is going to be a measure of what the energetics are, the strength of binding, if you want to say it that way, of an interaction between a ligand and a target. And that's going to be dependent upon the temperature, the enthalpy, which is the bond energy, and also entropy. So we can treat these non-covalent interactions as bonds. They're just non-covalent bonds. So this equation applies. So just to kind of get you a feel for this, we can talk a little bit about enthalpy. But in this case, I'm just going to go through just plain old chemistry where we actually are making and breaking bonds. So here we have what? Methane and oxygen getting together, and a reaction occurs that produces carbon dioxide and water. Okay? So you can measure the energy within every single bond in methane, for instance. Okay? And you can measure the energy between both oxygen atoms in dioxide, O2. So if you were to just calculate all of, and this is measured in kilojoules, if you just calculated the energy of all these bonds, here it is before the reaction occurs, and here it is after the reaction occurs. And you can see that there is less energy in the configuration of bonds in the products than there is in the reactants. That means that reaction is favorable. It can go forward, right? Because after that chemical reaction occurs, the resulting molecules are in an energetic state where the bond energies are lower. So that's enthalpy, okay? And it's using an example from regular chemistry where bonds are made and broken, right? But when you're talking about a drug-target interaction, we're interested in the bond energy not of covalent bonds, but of these non-covalent bonds, okay? And so the same equation can be applied. Entropy is a measure of disorder. That's the delta S term. So it's a measure of disorder. You could also think of it as the number of different ways molecules can be arranged. A good example of that, well, here is a volume with molecules in it. It starts out right here, and this is a very ordered state. But over time, those molecules are going to randomly distribute in this space, okay? So entropy has changed here. Here's a pile of bricks neatly stacked. Here is just a dumped out pile of bricks, much more disorder here. Here's a glass of ice. Here's a glass of water after the ice is melted. Which one do you think has more order? What do you guys think? Glass of ice or a glass of water? The ice, yeah. So, well, in this case, what is ice? It's a crystal, right? So they're going to be highly ordered there. So it really changes. But these are just kind of to get a feel for the entropy term. I also want to mention water molecules. And this is especially important in pharmacology. So here is a ligand, the green, blue, and red molecule here, sitting in the ligand binding pocket. But here's a little molecule of water stuck in the same pocket, okay? And it turns out that a lot of ligands get involved with water molecules when they bind to pockets. And that's called solvation. So water can facilitate the interactions because maybe one end of the water molecule hydrogen bonds with the target molecule, and one end of the water molecule forms hydrogen bonds with the drug. And so the presence of that water molecule can greatly increase the affinity, and that's called a solvation effect. Okay. So water can actually facilitate these interactions to facilitate association of a ligand and a target, but it may take a considerable amount of energy for dissociation to occur to remove that water molecule. And so that figures into things too. And you can have entropy considerations too, because a water molecule just floating around in a glass of water can move around, but once it is bound in this pocket in a particular way, it's a very ordered state. So you can get entropy involved as well. So this should get you thinking that ligand-target interactions can be described mathematically by things such as the Gibbs free energy formula, and we can actually measure these affinities. So we're going to talk about ligand-target affinities now. And what is an affinity? Let's say I have an affinity for chocolate ice cream. That means I like it, right? So an affinity is a propensity for two things to interact. And I like to interact with chocolate ice cream, actually. But, so a ligand-target affinity is basically what we're going to try to do. Affinity is, if you have a higher affinity, that means you like it more. If you have a lower affinity, you like it less. But what we're really talking about in pharmacology here is what is the strength of the interaction between the drug and its target receptor. And some drugs have very strong affinities, some have weaker affinities. And we want to find a way to actually measure that mathematically. And what that allows us to do is to compare one drug against another. Okay. So binding affinity. If you think about it, there's a very simple thing that goes on. The target in the presence of a drug can exist in two states. One is the target doesn't have the drug bound to it, and the other one is the target does have the drug bound to it. Okay? So here's a free target and a free drug. If they interact and the drug binds the target, now you have a drug-target complex. But remember, this can go in the other direction too. You can have a drug-target complex where the drug dissociates, and now you go back to the free drug and the free target. So this is happening dynamically all the time. There's always, in the real world, you have probably thousands or even tens of thousands, hundreds of thousands of targets in the body, maybe millions, and you have millions and millions of molecules of drug. So what's really happening on a molecular level is this association, and the dissociation is always occurring. But the real question is, what is the proportion of this state as compared to this state? I don't know what that is. Okay. So anyway, so we have association going this direction, the rightward arrow, dissociation going the other way, the leftward arrow. So let's give these some names. Let's call, for instance, the drug A and the target B. Association is A plus B goes to an AB complex, right? And so we can actually develop an equation called Ka, where we are measuring how much AB there is versus A plus B. And it's a very simple equation, and you should become familiar with this stuff, I think. So the association constant, Ka, is going to be a measure, a comparison of the concentration of AB, the drug target complex, to A plus B, which is A times B, which is the drug and the target free concentrations. So if you look at it, this is AB over here, and this is A plus B over here. The Ka is calculated by taking the concentration of the drug target complex divided by the concentration of free drug times the concentration of free target. So that's just a ratio, isn't it? It's the ratio of the drug and the target concentration where they got together divided by the free drug times the free target, okay? So if you think about that simple equation, the higher the number, the higher the affinity, right? The higher the association. So a high Ka for a drug means that drug binds the target very, very well. But we can also flip this around and look at dissociation on the left here. And this is where the drug target complex falls apart and now into free drug and free target. So here you have AB dissociating, and then you get A plus B, okay? And so this is called the equilibrium dissociation constant. And you'll see that we use the symbol Kd, which is the dissociation constant, to describe that. And Kd is equal to free A times free B divided by the concentration of AB. And anytime you see brackets in chemistry, it means concentration, okay? So in this case, the lower the number, the tighter the association, okay? Make sense? So Kd is one of the most important things you can know about a drug, the dissociation constant. And we could use Ka to measure it, but by convention, most drugs, we're talking about Kd. So if you know the Kd of a drug, you know a lot about the affinity of that drug for its target, okay? And the lower the Kd, the more affinity there is, the tighter the association, okay? And that's something you should just commit to memory. Low Kds mean tight binding, okay? Because of this. Okay. I do want to mention one other thing. Ka, there's a little bit of a nomenclature jargon issue between pharmacological chemistry and classic chemistry. And here it is. In pharmacology, Ka is an association constant, but don't be confused because in inorganic chemistry, for instance, Ka is an acid dissociation constant. So we're talking about two different things here. This has nothing, the pharmacology version of Ka has nothing to do with acids and bases. So we've probably all suffered through a lot of Ka stuff in chemistry, right? This is different. This Ka is different. There's another K constant that you encounter in pharmacology, which is called Ki. It is actually a type of dissociation constant, but it's used specifically for enzyme inhibitors, and so it's kind of another special thing. But remember, the Ka in pharmacology is different than inorganic chemistry, and really important, the Kd is how we measure the affinity of drugs for their targets. Okay. I can also mention something about this really quickly. Let's look at Kd. Bracketed A is a concentration, like micrograms per, or, a concentration. Bracket B is also a concentration. And the drug target complex is a concentration as well. And so concentration is measured in molarity, right? So what we have is, for instance, let's say we're talking micromoles. You do want to think in terms of how this works. You have molarity, millimolar, micromolar, nanomolar. Each one is 1,000 times greater, right? Let's say this is micromolars. So the units here would be micromoles times micromoles divided by micromoles. So micromoles times micromoles is micromoles squared, right? Divided by micromoles. Now you're back to just, so the unit of KD is going to be micromoles. So the KD units for KD are going to be a molarity value. Okay, so let's look at what this means. So here are a bunch of different KDs. Less than 10 nanomolar, less than 10 micromolar, less than 100 micromolar, greater than 100 micromolar. Okay. So these concentrations are all getting larger and larger, okay? But remember, with KD, if the value, the smaller it is, the tighter the affinity. We just talked about that, right? So the lower value of KD, the stronger the affinity. And that comes from that dissociation equation I just showed you. And so this is kind of a rule of thumb for how this works in pharmacology. If you have a drug that the KD is less than 10 nanomolar, that means very, very tight binding. Almost all of that drug is going to be associated with its target. Less than 10 micromolar, that's 1,000 times bigger, right? It's still tight binding. Less than 100 micromolar, moderate to tight binding. Greater than 100 micromolar, very weak binding. So many of the drugs in commercial use are in the very tight binding or the tight binding range for KD. Very few of them are like this because they just don't stick to the target. So get used to KD. It's one of the describing features of a drug for its target. And during drug development, if you have two molecules that both do the same thing, one has a very low KD and one has a higher KD. Probably the lower KD is the one you're going to use for commercial development because it sticks to the target better. You don't have to take as much of that drug, less likelihood of side effects because there's less free drug running around in the body. So it's a very important number you can calculate. So let's look at this a little bit from a kinetic viewpoint. Kinetic means concentration over time in this case, or binding. This is actually just no, this isn't time. This is just concentration versus the percent binding. And what we're doing is comparing how much drug binds to its target at different concentrations. So concentration is on the x-axis, and the percent of drug that's bound is on the y-axis here. And we're going to compare a high affinity ligand to a lower affinity ligand. So this one here, the red one, would have a lower KD. This would have a higher KD. And what you can see is as you increase the actual concentration of the drug that's introduced into the system, the high affinity one very rapidly occupies its target. The lower affinity one, you have to add tons of drug before it starts to even get up to like 60%, okay? And so, say that at 60% binding, you get a pharmacological effect, a favorable effect. It treats the disease. Well, for the red drug here, you'd only have to add a little bit to get up to 60. But for the green drug, you'd have to add 40. So we have, what, 5 compared to 40. You'd have to add, 8 times more of the drug, something like that. So molecules with higher binding affinities, which means lower KDs, they become bound sooner as you increase the concentration. Okay. So a little bit more about dissociation constants. I just want to give you a little flavor of how you actually determine this experimentally in the lab, so you know how these numbers are developed. And kind of a classic way to do this is a so-called filter binding assay, okay? So this is kind of an interesting thing. What you do is you take a filter, which is just a two-dimensional solid support. Could be like a piece of paper, could be nitrocellulose, some other thing like that. But it's something that protein sticks to, and you load up that protein with purified receptor, for instance. So you have a whole bunch of receptor immobilized on a piece of paper, basically, okay, a membrane. Okay. Then we want to find some way to measure the amount of ligand that sticks to that receptor. So first we fix up the receptor bound to the paper, and then we're going to add a bunch of ligand and see how much sticks. But the problem is you can't see it, right? This is submicroscopic. So what typically is done is you radio-label the drug itself. You just make an atom in that drug radioactive. And the nice thing about radioactive drugs is you can count them and measure them very precisely, okay? So you have radio-labeled drug, and you dump that on the filter that has the immobilized receptor, and some of it's going to stick, and some of it's not, right? And so you can then measure how much of that sticks by measuring the radioactivity, the radioligand, versus how much doesn't stick. It comes out in the wash buffer when you wash it off. So you can measure bound to unbound in that way. And so that's a very interesting way to do this, okay? So it's actually pretty simple. But to make this an even better assay, what you can do is actually perform the experiment many times well, anyway, that's how this works. But basically, you're mixing radio-labeled ligand with unlabeled protein, and you load a known amount of protein onto this membrane support. And then you let it adhere, and then you wash off the unbound ligand into the wash buffer, and you basically measure how much radiation is here versus how much radiation is out here. And the neat thing here is you can measure A, B, and AB. So if A is the drug, remember that's radio-labeled. B is the protein, and we know how much we put on there, so we have a concentration. So we already know B just because of how we assembled the filter receptor complex. We can then get the concentrations of the drug filter AB here, because we measure how much radiation sticks, and then the washed off stuff that doesn't stick, we measure that, okay? And so you can actually calculate KD with such an experiment, right? Okay. There's one other kind of consideration, and has to do with competitive binding experiments. So this one here just had hot ligand, and that was it, radioactive ligand. What if we did a similar experiment and we used a single concentration of hot ligand, but we repeated the experiment many times, and we started adding in more and more unlabeled, unradio-labeled ligand. So we might do the experiment like 10 times. First one has no unlabeled ligand. The second one has twice as much. The next one has 4 times as much, then 8 times. So you do a different series of concentrations of the cold ligand, okay? And then you can see what happens. So the neat thing about radioactively labeled drugs or any compound is the radio-labeled drug is chemically identical to the unlabeled drug. It just has a radioactive atom in it. But chemically, it's going to be the same. So as you start adding more and more cold ligand, that means unradio-labeled, you're going to have more competition going on. So there's a competition for binding of the radio-labeled ligand and the unradio-labeled ligand. and they both have the same affinity because radiolabeling doesn't change affinities. And the more cold ligand you add, basically the less signal is retained on the membrane support because if you have 100 times as much uncold ligand, 99 out of 100 times the receptor is going to bind the cold one. It's going to choose the cold unlabeled ligand. And so you can then actually quantitate this, and you can see that as you add more and more cold ligand, less radiolabeled ligand is retained. And so these competition experiments with a mixture of radiolabeled and unlabeled ligand are a great way to develop KDs, and it's even more accurate. And you can develop this term called IC50. That's the concentration of a drug that blocks 50% of the binding to a known standard. So this is kind of interesting because these types of competition experiments are very the was radiolabeled and unradiolabeled. But you can do competition experiments where you add a drug and then you add an endogenous ligand, like estradiol and estrogen, for instance, right? And you could see by doing such experiments which one has the higher affinity, is the estrogen or the estradiol. So these competition type experiments are used widely in science, and you can get quantitative numerical readouts of affinities. And here is an equation that will not appear on the exam. It's the only one that I talked about that might not appear on the exam, but it gives you an idea. Here's that IC50, which we just talked about, concentration of drug that blocks 50%. It's a 50, it's a concentration that gives you 50% competition here. And you can develop through this equation something called Ki, but basically, you don't really need to. I'm not going to give you a problem to try to figure this kind of stuff out. But Ki is the dissociation constant for the unlabeled drug in the absence of a competitive binder. And so you can actually, once you get that IC50 value, you can plug it into one of these equations and figure out what Ki is. So this Ki is going to be a property of the experiment, but the Kd is a known dissociation constant for the drug. And so I just wanted to show you this because this is what happens in pharmacological labs when they're characterizing new drugs and they want to find out which one binds most strongly. You remember structure-activity relationships? We were talking about that last time. Oftentimes when you change in the pharmaceutical industry or in academia, you start out with a drug. It's a known chemical entity, but typically the first one you get isn't very good. Maybe the Kd is only like millimolar range or something like that. Well, can you make chemical changes to that drug so that you increase the affinity of that drug for its target? And you could take a molecule that has promise and turn it into a molecule that actually works by changing the structure of the drug. And so that structure-activity relationship describes a lot of things, but one of the things we're really interested in is, can you alter the chemistry of the drug so that the affinity is increased? That means the Kd goes down, and you can take a molecule that would never be usable as a drug and turn it into a molecule that now is usable for a drug because you tweaked its chemistry to make its affinity higher. And the way you actually do that in the lab is with filter binding experiments. That's basically an assay that gives you a numerical readout of how strong a drug binds to its target. Okay, so you now have kind of the very basics of how we do this mathematically, and we can talk about things in terms of numbers. So on Friday, there's going to be a bit of a challenge. We're going to talk about selectivity. And that is, you have a whole bunch of drugs, a whole bunch of related targets. Which ones are most selective? It's like a bag of M&Ms with a bunch of colors. One person might only like the red ones. Somebody else doesn't care. They just eat any of them. The person who eats the red ones is a selective person, right? We want our drugs to be selective in general. So we'll get into that. We know how to use numbers to talk about it. And that's it for today. Thanks.